everything. Very little details have shown up within the exam. Some of the big things do pop up where you see primary differences between each of the types of methods. So we've got, as far as a bulk review, what was our first chapter about?
DNA.
Kind of.
DNA 9.
We're looking at structure and ultimately structure of kind of DNA, sort of, a little bit of structure on RNA. We're really concerned about the structure of our nucleotides. Okay? How do they come together to make different forms? We see some DNA things, supercoiling, all that kind of fun stuff. Then what did we move to?
Replication.
We had replication, which was what? Right? Just duplicating our DNA, duplicating our genetic information. We didn't talk about it, and it's not on the test, but there is reverse replication. No, reverse transcription, sorry. We should move to transcription as the next one. So the next method that we talked about was our transcription. What's happening in transcription? We take our DNA and turn it into RNA. There is something as reverse transcription, which is going to be RNA to DNA, so way to pick all the examples of where it happens and not target what it is, which is true. I mean, you did a good job. But we're looking at the reverse process, so RNA to DNA. So you can see that happen in a lot of different cases. You mentioned viruses. We don't do it with bacteria, do we? But we see it primarily with viruses. Where else do we see it that we did actually talk about? And Jonathan, maybe you can jump in on this one.
Oh,
Telomeres.
telomeres, yeah.
Telomeres, you're looking at a telomerase with an RNA sequence that it then uses to code for the telomere sequence on our DNA strands. Okay, so we do see a reverse transcription in that case with our telomeres because we're coding from RNA to DNA. And then the last process, that's our translation. We're going RNA to our proteins. Within each of these sections, it breaks down on the exam roughly 8 questions per section. Okay. For those 4 sections. Okay. And it's like 6 or 7 questions each, multiple choice, and then 1 short answer for each of those individual sections of material.
So there's only 30 prokaryotic, eukaryotic differences.
There are 30 multiple choice, and then there are, I think that was right, 6 short answer. Maybe 5. I don't remember. 5 or 6 short answer. Okay. And these short answer, except for like maybe 2, are short answer. Like I could write a 1-word answer for a good 2 or 3 of those short answers. Yes. They just weren't really available to do multiple choice questions on. So some of them are adding arrows, mechanistic arrows, to an individual reaction, identifying nucleophile, electrophile kind of ideas between those reactions. So structure. What questions could pop up about structure for DNA, RNA?
The base pairs.
So we could push all the way to base pairs, but let's take it simpler. Simpler?
Okay.
More simple?
Simplify it.
Differences between DNA and RNA, I would say, is still too complicated because you're now still at a complex molecule of DNA and RNA. Go even easier. What makes up DNA? What makes up nucleotides?
Deoxyribose.
Nucleosides. What makes up nucleosides?
Deoxyribose.
A sugar and the base. Okay. So your amino group. What bases do we have? Acid base. No. Add some base, okay. Drop the base. What bases do we have? Okay, I can accept that. A, C, T, G. Does that apply to RNA as well? So I think I heard it also. We also have U. Can we classify those? We have purines, and we have the pyrimidines. Okay, your purines have how many rings? We got 2 rings. The pyrimidines, 1 ring. Where do our nucleotides fall within these? I kind of don't like this question, but it's kind of important. Where do the nucleotides fall? So what is A? Purine, pyrimidine. What is T? Okay, so that's nice. What's the base pair? AT, right? So if you know what one of them is, you should know what the other one is. Why is A a really nice one to know? Because we can get away with one more base pair. What's the other base pair? Our uracil, which is then also going to have to be a pyrimidine. What's our other partner? G and C. Which one's which? G is the purine. I'm really just doing that because you all said it was. And C. I won't ask about inosine. So if you want to memorize that, feel free to, but there are no questions about inosine. Or the pseudo stuff.
If you're not going to do the inosine, then we don't have to understand the wobble.
You aren't responsible for knowing the base pairing in the wobble, but are you responsible for understanding what wobble does?
Okay, yeah.
Absolutely. Where does the wobble pairing show up?
First, when he gets drunk.
In these 4 categories, where does wobble base pairing appear?
Translation.
I see what you're answering. We are not looking at the nucleotides, we're looking at the bulk red things. The bulk red things, the wobble pairing is all about translation. Is that right? So what does wobble pairing do for you?
Increases the potential to add,
And decreases the amount of tRNAs.
Decreases the issue for mutations. So if mutations pop up, it's not going to be as big of an issue because it's going to still code for the same thing. So if you have AUC versus AUT,
oh, I thought she said
okay, so it minimizes mutation issues. What else can it do for you?
Decreases the variety of tRNAs.
We have less tRNAs that we would actually need to code for. So we don't need 64 tRNAs. We can get away with a few less than that because that wobble base doesn't have to be perfect.
So it conserves tRNA?
Okay, so it will conserve the amount of tRNA.
Is that it?
About what? Wobbles
For
or
wobble.
everything?
No, for wobble?
For wobble, that's
So those
pretty
are the-
reasonable.
the 2 things? Okay.
What's next within our structure that we could look at? Oh, actually, we already mentioned some of those. Our base goes up to nucleosides, which means, what's the difference between a nucleoside and a base? One has a sugar. What's the sugar? Ribose for RNA and deoxyribose for DNA. What's the difference between those 2 ribose structures? You're looking at the second carbon
Having an H instead of...
having H's instead of the OH. What's the sequence of the OHs as far as up, down, all the way around?
Oh,
Beta is correct, but that doesn't help you on which way it goes.
I don't know. Oh,
Up, down, up.
I think that is correct. So it'll go up, down, up for your sugars if you start right to left within the Hayworth projection. If we look at the next tier up, what's the next tier up from nucleosides? Nucleotides, what's the addition there? We added the phosphate group. Okay. After that, we've now got our nucleotides. We went through the base pairing, which we already kind of talked about. A goes to T or U, depending on whether you're looking at DNA or RNA. C goes to G. Okay. We could then look at DNA structure. And in DNA structure, what do we have? DNA, thank you. We have a double helix, which means for every A there is an equal amount of T. Okay, for every G there's an equal amount of C. So we got a double helix. What else can you tell me about the secondary structure of DNA?
You have a wide groove and a narrow groove.
Yes-ish, but before we even get into the major versus minor grooves, you have B,
Oh, Z.
A, and Z, right?
Yeah, BZ.
B is really where you're getting your major and minor, for lack of a better description on that. Okay. Which of these is the most important form physiologically? B. That's the most common, most stable form. What else is a characteristic of B?
Right.
It's a right-handed helix. A is also officially a right-handed helix. A has a tighter coil, I thought. Yeah.
Because it's just pretty much like two minor groups, right?
Yeah, I think so. Other big thing that I would drop under the B DNA.
Hydrophobic, hydrophilic.
We do have hydrophobic interactions primarily. We have base stacking. Okay, so when we look at how bases align up, we typically think about the hydrogen bonding. But the second important aspect of that is not just the hydrogen bonding, but the
Pi bond.
the what?
The propeller twist or something.
So it has to do with the propeller twist. Within those, we don't get even flat stacking. There's a slight rotation to them. Okay, which does what?
It makes the hydrophobic have more interaction.
It maximizes a hydrophobic stacking and minimizes the hydrophilic section for access to water. Okay. The hydrophobic stacking that's important is what I think you were trying to allude to, Braden, the pi stacking. Okay. That base stacking interaction is really, really important for the stability of the DNA because we get those kind of stacked p orbitals that stabilizes that interaction. Oh, inner diameter of the B helix? Inner. The inner was about 11 angstroms, which matches for that hydrogen bonding distance. Okay. For some reason, I think that's important, but I can't remember why. I don't think I gave that exact number, but I think I did ask a question about that inner diameter. Okay. The rest of them, I'm pretty sure I didn't ask any questions about, which I reason away as being reasonable because they aren't physiological. Major, minor, so capital M for major, lowercase for minor. What else came out of that chapter?
Conserving.
Supercoiling.
Yeah,
Conservative versus semi-conservative versus all of that is in reference to replication. That is a very important topic. What experiment proved our semi-conservative? Whatever those guys' names were. They started with heavy DNA, okay, made a whole bunch of, or grew a bunch of cells in really heavy DNA. How is it heavy? They use a heavier isotope of nitrogen. They then changed the medium and let it propagate outwards, and then looked at the density of the DNA per generation. Okay, and what they noticed was that only 2 cells ever held heavy DNA in them, and it was a hybrid of heavy and light, which meant that one strand of your heavy DNA was mixed with a light strand every single time. The rest of it was all light, which meant that you had semi-conservative because you kept a strand. What would have been completely conservative? If one cell maintained heavy, and the rest were all light. What if it was not conservative at all?
Mixed.
You're going to get a massive mix of everything. Okay? Heavy is going to show up everywhere because what you're doing is decomposing that heavy DNA to make new strands of DNA. Okay, so nothing was conserved, you destroyed everything. Okay, so we got semi-conservative DNA replication. What's the issue with our supercoiling? So gyrase, I don't know why I switched the color, but too late now. Gyrase was used for helping with our supercoiling. What types of supercoiling did we have?
Negative.
We have negative and positive. What happened in each of those cases? If we do negative supercoiling, what happens to the amount of bases per turn? You increase the amount of bases. Okay? Because what you've done was taken that coil and you've now unwound the coil. Okay? So we're looking at an unwound section of DNA. Is that actually unwound or is that unwound? That's good. Positive is going to be overwound. When would you want negative or positive?
You want negative when you're actually doing transcription.
We would want the negative when we're doing transcription and replication. When would we want the positive? When we're doing nothing to it. We want to keep the DNA protected. So we'll supercoil it to actually compact everything down nice and tight and prevent anything from getting in and interacting with it.
In the positive, you have like more histones.
Sure, I don't know.
So the system's not on there.
No, there's no questions on histones. Or hats or whatever those are.
No hat.
Replication. What else is going on in our replication? What are our steps of replication?
The origin.
So we're going to have some kind of initiation, which is going to start at an origin.
Multiple origins in eukaryotic, one in prokaryotic.
So prokaryotes we have one, and eukaryotes we have multiple. Multiple, or oh, here we go. You'll like this. Let's use the standard biochemistry nomenclature. P for prokaryotes have one origin. Get it? E for eukaryotes have multiple.
I like it.
Fine, I thought it was funny. What else do we have to worry about in our initiation?
That it moves in two directions.
It's bidirectional in both cases, meaning that our replication is going to continue outwards both ways. Bidirectional means that it's going outwards from a center point, so it goes out here and here. Which way does our replication run, though?
5'.
It always runs 5' to 3'. So it's 5' to 3', but it is bidirectional. Okay. I don't think I asked anything about specific things. Thank you. That is an important thing to address in here. We have parts of our probably classify that as elongation. We're looking at synthesizing. We'll have a leading strand, which will be just standard, easy to go. A leading, which we'll call, I guess, continuous. The lagging strand, fragmented. Ooh, I like that word. Fragmented. And this is where we're going to get our Okazaki fragments. Okazaki fragments. Okay, coming in with this. So how do we get each of these things to start, though, both the leading and lagging strands? We have to have an RNA primer, which would be catalyzed in there by a DNA primase, I think is right, to actually get that to bind. Once the primer is in there, then we can start adding nucleotides on. So on the leading strand, we only have one RNA primer because we're freeing up an immediate nucleotide that can immediately be slid in there. In the lagging strand, we're going to run into an issue because we have to run 5' and 3', but we're exposing the 5' end each time. Okay, so we can't synthesize appropriately. So that's why when we look at the lagging strand, we'll put on multiple primers for each of those sections. We'll then polymerize in the proper direction, but then what happens is our polymerization comes back and it butts up into the next RNA primer that was already put in there. So now the polymerase freaks out, doesn't know what to do, and it spits it off. And we have these sections of replicated DNA.